On this week's GARNet Research Roundup, I'm really delighted to be joined by Charles Melnyk, who is currently working at the Swedish University of Agricultural Sciences in Uppsala. But we're going to talk about some work that he did when he was at the Sainsbury Lab at the University of Cambridge. And this work is just published in PNAS, the title of which is Transcriptome Dynamics at Arabidopsis graft junctions reveal an intertissue recognition mechanism that activates vascular regeneration. So Charles is really a world expert in grafting and the molecular changes that happen in graft junctions. So thanks very much, Charles, for joining us today, and it'd be great if you can give us an overview of the work you did in this paper.
Well, thank you very much for the invitation to speak here. Yeah, so I mean, I've been interested in grafting for probably well over 10 years. And I mean, it's an incredibly important horticultural and agricultural technique. I mean, grafting is used worldwide for fruit trees, for grapevines, tomatoes, a lot of very economically important species. But we actually know very little about how plants graft. And I think it's quite a fascinating process that you can cut plant in half, and you can either stick it to a completely different individual, or even in some cases, somewhat related species, and then these tissues will fuse, and then everything within a number of days or weeks will reconnect, and you form this very nice example of a chimera. Mm-hmm. So the point of this paper was really to try to understand the mechanism behind graft formation, and using a transcriptome analysis. So we did we grafted a huge number of plants. We harvested tissue graft junctions, and we wanted to see genes that were being differentially expressed. Mm-hmm. And we were very fortunate that, of course, there was thousands of genes that were differentially expressed, and we could actually observe a lot of the classic healing processes, regeneration processes were being activated. Things associated with vascular formation, so cambium, xylem, and phloem genes were all being activated, which was very nice. It was very reassuring to see this, that we could see this with the species that we were working with, which is the Arabidopsis hypocaudal tissue that's being grafted to another Arabidopsis hypocaudal.
I was going to ask you initially about the experimental design, actually. So we know that grafting is not a 100% successful occurrence. So how do you overcome the inherent variation that occurs? So some of the plants won't survive the graft. So how do you overcome that within your experimental design?
Yeah, I mean, it's certainly when I first started grafting, the success rates were extremely low, mm-hmm. And I think it was a lot of trial and error and trying different techniques with light, humidity, temperature to really try to get it up to a really good success rate. Mm-hmm. So I mean, at the moment, success rates are probably around 90%. Yeah, okay. Okay. But you're right. I mean, it's still there's a percentage of plants that essentially will fail to graft. And in our transcriptome analysis, there wasn't much we could do about that. So especially at the early time points where we really don't know how things are going to work. Mm-hmm. But a good indicator is just how well the plants are lined up, even after 24 hours, where you see things starting to fall apart. Then those ones were generally removed because they weren't going to graft successfusly. And then later on, when things fell apart, then we would throw them out. We wouldn't use them for the analysis.
I see. And you see in the experimental design, you showed that you use multiple plants each time, and if you think you have a 90% success rate across all, then it will even itself out.
That's right. I think I worked out we grafted over 1,000 individuals for this paper. So I mean, it took me months of grafting to get enough samples and materials. But you're right, we didn't do a single graft junction. I mean, all the time points had at least 50 individuals that were pooled together just to get enough material, because the Arabidopsis hypocaudal is only a couple hundred microns across. If we were using, tomato plants, or we were using, trees, then I could have done, just one or two grafts, and it would have been technically far easier.
So can you tell us something about the gene expression changes you saw at either side of the junction?
Well, that's right. I mean, we did this massive transcriptome analysis. I mean, we had we had 3 different treatments, 5 different tissues. We had 82 libraries that were being made, and we saw approximately half of the genome was being differentially expressed. Okay. So, at least under one condition. I think what's happening here is when you cut the plant in half, basically the whole thing is gonna freak out. You're having a massive wound response. So, we basically come to the conclusion that half of the genome is being differentially expressed, but how can you really get interesting and informative biological information from this? Yeah. And I think that the real eye-opening moment is that when we realized that there was actually a lot of genes that were basically being asymmetrically expressed at the junction. So they were being more highly expressed on one side than the other. Okay. And those are actually a lot of the genes were associated with that. And then we later went on to discover why there was this, we think, this inherent asymmetry, why some things are activating first on one side than the other. So the tissues were not behaving the same on either side. But then what was also very interesting is there actually were a lot of genes that were behaving identically on both sides of the graft junction. So they were both activating very, very rapidly. And we think this is coming down to some sort of underlying recognition mechanism. So there's something in the bottom of the graft that activates very rapidly in the contact of the upper part of the graft.
Okay.
So we don't see this in simply a situation where we cut the tissues and we don't graft them. We don't seem to have this rapid gene activation below the cut site. You need the contact for this to occur.
And through the data, it looks as though you can kind of pinpoint that to a specific action of a particular hormone.
Yeah, so the strongest evidence, we looked at all the phytohormones to see where the response associated with those hormones overlapped with our transcriptome data set. And the strongest evidence by far is for auxin, which of course is the plant hormone that seems to control nearly everything. So, perhaps it wasn't that surprising that we discovered that auxin was playing some role in this process. And certainly a lot of the auxin transporters are being very highly expressed around the graft junction. Okay. So it looks like, after the cutting process, we get upregulation of the transporters. Auxin's probably getting pumped across. And a lot of the very early activators, what we think are these early activating recognition genes, at least one of them we tested, and it looks like it's very auxin responsive. Okay. So within 24 hours of treatment with auxin, it's getting very nicely increased in expression. Okay. So at least our data is extremely consistent with, even though we've cut the plants in half, the vascular tissue is severed, there should be no real transport through the phloem or xylem, but still we have this recognition response that we think is being generated by the movement of auxin from the shoot to the root.
So what you're suggesting is that there's this cell-to-cell transport which is important at the graft junction that for establishing the graft, I guess.
That's right. I mean, there's some evidence. There's a bit of auxin in the phloem, but actually the majority of auxin is being transported from cell to cell.
Okay.
We think through the xylem parenchyma, perhaps also the cambium. So I think what we observed was actually entirely consistent, that there's perhaps this cell-to-cell transport that we know is happening in ungrafted individuals. In our grafted individuals, even though we've cut the vascular tissue, it is not functioning for the first 3 days. It doesn't seem to matter. We can still get the cell-to-cell transport happening across the graft junction.
Yeah, okay, so that is striking. That you talk quite a lot about obviously sugar responsive genes in the paper, but it's clear that the auxin response is coming on even before that sugar transport would have occurred.
Absolutely,
Yeah,
that's right. So, like you say, I mean, we think this asymmetry. So these genes that are more highly expressed on one side than the other, those really correlate quite nicely with things that are sugar responsive. And that makes a lot of sense because our phloem, you do the grafting process, you cut it through the middle. And of course, it's going to take 3 or 4 days for the phloem to reconnect, and at that point, the sugars are not going to be able to transport until our phloem is reconnected.
Yeah. And on the sugars, one interesting little bit of data in the paper, you showed that when you put the grafted individuals on low concentrations of sugar, you actually decrease the successfulness of the graft. Is that right?
That's right. Yeah, okay. Exactly. Yeah. Okay. So it's an interesting observation. I mean, we couldn't completely block graft formation, but we can reduce its efficiency by giving very, very low levels of sugar, suggesting that it could be that perhaps this low levels of sugar are slightly stressful for the plant. This idea that maybe you need this asymmetry in sugar, you actually need a higher concentration on one side than the other to maybe activate some of this regeneration process or the healing process. And then when we just simply coat the whole plant in sugar, we lose this ability, and somehow this is perturbing graft formation.
Yeah, the tissue sections are quite happy in their own sucrose. They don't need it to supply that, I suppose, in that way. So, as I mentioned, about a year ago now, you moved from Cambridge to Uppsala. So, can you talk about a little bit about the work that you're continuing in this area? Because I know you're still focusing on the graft work.
That's right, exactly. So, I mean, I've brought a lot of this grafting work with me here, and we did this transcriptome analysis. We discovered thousands of genes are being differentially expressed at the graft junction. Mm-hmm. So we're really trying to identify those genes that we think are most important for graft formation. Mm-hmm. And we have some very nice candidate genes. We've been focusing on transcription factors, and it's really trying to modify these genes to see if we can inhibit or improve graft formation. We're using Arabidopsis, but we're also now starting with some other species. We work on rice. We also work on corn. So that's one aspect of the project. And actually, the other half of the lab is working on parasitic plants.
Okay.
Which I think is also horticulture, very important. I think it's also fascinating that you can get these two plants, very different species, the parasite and the host, to come in close contact, tissue fusion, and also form vascular connections between them. So it's analogous to a natural grafting situation. But how much mechanistic similarity there is between grafting and plant parasitism is completely unknown.
Okay.
But our lab is looking for those parallels, and we have people who are also looking for genes important for parasitism.
Okay. It's extremely translational, this work, obviously, to move from Arabidopsis into other plants. But maybe this is a slightly off-the-wall question. Have you interacted with anyone who works in the mammalian field to see if there's any analogy with, grafts in animals and about vascularization of grafted tissue and things like that? Is there any analogy there, or is that totally crazy?
Yeah, no, I think it's a really good question. I think it's really interesting, and I get it occasionally.
Okay.
When I give talks, and I'm often, it's a tough one to answer. I mean, I think with mammalian grafts, I mean, you have this really nice defense response that gets activated. I mean, you basically, you have to give drugs and chemicals to try to reduce this defense response, where the immune system is basically attacking the various tissues. Yeah. And I think at least with plant grafting, I think we're also seeing a defense response.
Okay.
I think when we're just grafting Arabidopsis thaliana hypocotyls to themselves, even though it's different plants, I think genetically the plants are identical. But the real interest of horticultural grafting is grafting divergent species and combining the best traits of each plant together. Yeah. And I think a lot of reason why grafts are not working is because we're getting inappropriate immune responses, we're getting inappropriate defense responses.
Okay, cool.
So maybe on a very broad term, we could start looking at, what's happening in mammalian systems and trying to understand how can we, modify these defense responses to allow grafts to take, and also understand how parasitic plants can somehow evade these responses to allow them to infect really, really divergent species.
So thank you very much, Charles. We just scratched the surface of the enormous amount of data which is in the paper, and all the data is available online as well. So I'd encourage people to look at it. There's a lot of novel genes that we haven't mentioned here. So thanks very much, Charles, for chatting about this, and we look forward to hearing about your future work in this area. That's great.
Okay. Thank you very much.